Hi, my name is Rui Chen. I'm currently a professor at Department of Ophthalmology and also the Center for Translational Vision Research at UCI. Uh, I have been working on, my interest is in genetics, particularly the genetic factors that affect the vision development and diseases. Inherited retinal diseases is a rare form but severe form of the retinal degeneration disease. It affects about 1 in 3,000 people, and they are not reversible. And currently, the exciting part is there's a gene therapy have been developed to treat those disease. So we have been particularly interested in identifying mutation that cause this disease so that the patient can be then identified and mapped to the clinical treatment and clinical trial ongoing. The way we do it is we form a large international collaboration that together we have performed molecular screening for over 8,000 patients. And among them, we already identify mutations in more than 7,000 of them. And so those patients have a chance to be moved to the next stage to get treated. To identify those genes, as you know, the genome are 3 billion bases. So often the mutation is really only involved in one base. So it's really hard to find it. So to do that, we perform next generation sequencing so that these days we can cover the entire genome. And we left about 3 million variants we need to sort out, which is the mutation. So we use a variety of tools, both informatics and experimental, to basically narrow down and identify the high priority changes. And so that we can then do additional follow-up study, such as genetic segregation, model system, modeling, and biochemistry, molecular biology to identify the mutation. So that's the technology we use. In the last 5 years, we have been very excited about this new technology called single-cell omics. Basically, what you can do is you can perform omics level, genome level, transcriptome level for individual cell. So as a result of this technology, now we have opportunity to do two things. One is for the first time, we can identify all cell types in a tissue so that we can then link the individual cell type to its function to the disease. But I think more exciting is actually you can now do millions of experiments in one tube, right? That dramatically increase your throughput and also reduce the cost by 1 million times. So use this method because as I mentioned earlier, each people have millions of variants in their genome. To understand their function, if you do one by one, it will take forever. But with this single cell omics technology, it is now possible to perform high throughput functional assay at a reasonable cost and reasonable time frame. So that's why we initially very interested in single cell technology. During the course, we have been using this technology to identify all cell types and draw and generating the single cell atlas of our human eye so that we know what cells are at play here. But in the meantime, also identify variants across many, many patients or people, and then look at their consequence in the individual cell type context in terms of the function. And then we also develop a method so we can perform high throughput function assay use the single cell omics technology. Yeah, I think we are really at a very exciting time. So, for many more reasons, but I think to me is really driven by the technology development. So as I mentioned, the single cell omics technology that you can do experiment 1 million times faster and 1 million times cheaper. But also in the last 20 years, because the next generation sequencing technology, human genome projects, so on and so forth, for the first time, we can look at our own genome at very low cost, few $100 to look at all your genetic code of yourself. So combine these two technology, and also need to mention the AI, the machine learning, right? So if you take this big data and then you feed on into the proper machine learning model, then it can help you to interpret and prioritize the causal mutation underlying the disease and biology in general. So I think in the future, it's really combine the big data, AI, high throughput functions, and then we form this very nice feedback loop. So we can rapidly identify disease-causing mutation, and then we can design therapeutical method to treat the patient. So I think that's where we're heading now, and then we are actually accelerating on that path. And then this is what's what happened basically. It's happening right now.